on lesson 9 of science, that is Heredity and Evolution. For grade 10, we discussed about inheritance of traits, evolution and classification. So, in continuation to this, today we will discuss about how life originated on earth. In addition to this, we will discuss that how the complex life that we see today has evolved from the simpler ones. So, dear children, let us begin with our today's session. Now, we will discuss about how life originated on earth. Children, the theory of evolution given by Sir Charles Darwin tells us how life evolved from simple to more complex forms, and Mendel's experiments gave us the mechanism for the inheritance of traits from one generation to the next. But neither of the two could tell us about how life began on the earth. It was cbse Haldane, a British scientist who in 1929 suggested that life must have originated from the simple inorganic molecules which were present on the earth soon after it was formed. He speculated that the conditions on the earth at that time were far different from the conditions we see today. Maybe the initial environmental conditions could have given rise to more complex organic molecules that were necessary for life. According to him, the first primitive organisms would have arisen from further chemical synthesis. But then the question is that how did these organic molecules arise? An answer to this was suggested by the experiment conducted by Stanley L. Miller and Harold C. Urey in 1953. They assembled an atmosphere similar to that thought to exist on early Earth. This had molecules like ammonia, methane and hydrogen sulfide, but no oxygen, and the temperature was maintained just below 100 degrees Celsius. Sparks were passed through the mixture of gases to stimulate lightning. At the end of a week, 15% of the carbon had been converted to simple compounds of carbon, including amino acids. These amino acids can make up protein molecules and hence explain the origin of life on the earth. Evolution and classification. Based on the theory of evolution, we can work out the evolutionary relationship of the species we see around us. To understand this clearly, we will have to go backwards in time. We can do this by identifying hierarchies of characteristics between different species. For this, we will have to group organisms with similarities in one group. Then, we can try to study their characteristics and see which characteristics decide more fundamental differences among organisms and which ones decide less basic differences. Now, what is meant by characteristics? Characteristics are details of appearance or behavior. In other words, a particular form or a particular function. Plants can do photosynthesis. This is also a characteristic. Cell is the basic unit of life in all organisms. A basic characteristic of cell design that differs among different organisms is whether the cell has a nucleus or not. Among organisms with the nucleated cells, which ones are unicellular and which ones are multicellular. This property marks a basic difference in body design because of specialization of cell types and tissues. Among multicellular organisms, whether they can photosynthesize or not is the next level of classification. And now, among the multicellular organisms that cannot photosynthesize, whether the skeleton is inside the body or around the body will mark another fundamental design difference. We can see that even in these few questions that we have asked here, a hierarchy is developing which allows us to make classification groups. The more characteristics two species will have in common, the more closely they will be related. The more recently they have had a common ancestor. For example, a brother and a sister are very closely related. They have common ancestors in the first generation before them, that is their parents. A girl and her first cousin are also related, but less than the girl and her brother. This is because cousins have common ancestors, that is their grandparents, in the second generation before them, not in the first one. Children, just now we have discussed how life originated on earth from very simple forms and then evolved to the complex structures that we see today. In our next topic, that is tracing evolutionary relationships, we will try to find evidences that prove how we all have evolved from a common ancestry. When we try to follow evolutionary relationships, how do we identify characteristics as common? These characteristics in different organisms would be similar because they are inherited from a common ancestor. Consider this fact as an example that birds, reptiles and amphibians all have 4 limbs, as do the mammals have. The basic structure of the limbs is similar, though it has been modified to perform different functions in various vertebrates. Such a homologous characteristics helps to identify an evolutionary relationship between apparently different species. However, all similarities simply in organ shape are not necessarily because of common ancestry. What would you think about the wings of birds and bats? We know that birds and bats have wings, but squirrels and lizards do not. So, are birds and bats more closely related to each other than to squirrels or lizards? Before we jump to this conclusion, let us look at their wings more closely. When we do this, we find that the wings of bats are skin folds stretched mainly between elongated fingers, but the wings of birds are a feathery covering all along the forearm. The designs of the two wings, their structure and components are thus very different. They look similar because they have a common use for flying, but their origins are not common. Evolution. This makes them analogous characteristics rather than homologous characteristics. Children, next we will study about fossils. How do we know that these extinct species ever existed? Yes, children, we know this from finding fossils. Then what are fossils? Usually, when organisms die, their bodies will decompose and be lost. But every once in a while, the body, or at least some parts, may enter in an environment that does not let it decompose completely. For example, if a dead insect gets caught in hot humid mud, it will not decompose quickly, and the mud will eventually harden and retain the impression of the body parts of the insects. All such preserved traces are called fossils. How do we know how old the fossils are? There are two components to this estimation. First method, if we dig into the earth and start finding fossils, it is reasonable to suppose that the fossils we find closer to the surface are more recent than the fossils we find in deeper layers. The second method is fossil dating. This is done by detecting the ratios of different isotopes of the same element in the fossils. How do fossils form layer by layer? Let us start 100 million years ago. Just imagine when some invertebrates died. They got buried in the sand. With the passage of time, more sand accumulated and due to high pressure formed sandstone. Millions of years later, dinosaurs living in the area also died, and their bodies too got buried in the mud. This mud also got compressed into rock above the rock containing the earlier invertebrate fossils. Again, millions of years later, the bodies of horse-like creatures also died in the area and were fossilized in the rocks above the earlier rocks. Much later, by erosion or by water flow, the mud got veered away from some of the rocks and exposed the fossils. As we dig deeper, we found older and older fossils. Evolution by stages. Children, a question that arises here is, if complicated organs such as the eye are selected for the advantage they provide, then how can they be generated by a single DNA change? Surely, such complex organs must have been created bit by bit over generations. But a point on which we should think over here is that how intermediate changes are selected. For this, there are a number of possible explanations. An intermediate stage, such as a rudimentary eye, can be useful to some extent. This might be enough to give a fitness advantage. In fact, the eye, like the wings, seems to be a very popular adaptation. Insects have them, so does an octopus, and so do the vertebrates. And the structure of the eye in each of these organisms is different, enough for them to have separate evolutionary origins. A change that is useful for one function to start with can become useful later for quite a different functions. For example, some dinosaurs had feathers which provided insulation to them in cold weather. But they could not fly using these feathers. Later, the feathers became useful for flight in birds. Dinosaurs were reptiles. So, from the above discussion, we conclude that birds are very closely related to reptiles. Or in other words, very dissimilar looking structures evolved from a common ancestral design. It is true that analysis of the organ structure in fossils allow us to make estimates of how far back evolutionary relationships can go. A good example of this is a wild cabbage plant. Humans have, over more than 2000 years, cultivated this as a food plant and generated different vegetables from it by selection. This is, of course, artificial selection rather than natural selection. Some farmers wanted to reduce the distance between its leaves, and they bred the cabbage we eat today. Some farmers wanted to select for arrested flower development. This led to the development of broccoli, and cauliflower developed from the sterile flowers. Some selected for swollen parts and came up with kohlrabi. Some simply selected larger leaves and came up with a leafy vegetable called kale. If such kind of selections would have not been done by we the humans, then can we even think that all these structures can descend from the same ancestors? Another way of tracing evolutionary relationships depend on the idea of changes in DNA during reproduction. If that is the case, then comparing the DNA of different species should give us a direct estimate of how much the DNA has changed during the formation of these species. Molecular phylogeny. Just think that the changes in the DNA during cell division would lead to changes in the proteins that are made from this new DNA. These changes would accumulate from one generation to the next. Molecular phylogeny helps us to trace the changes in DNA backwards in time and find out where each change diverged from the other. Children, now let us quickly revise whatever we have learned in this chapter today. Today we have learned that variations arising during the process of reproduction can be inherited. Sexually reproducing individuals have two copies of genes for the same trait. The trait that gets expressed is called the dominant trait and the other is called the recessive trait. Number of chromosomes in each generation remains maintained. Traits in one individual are inherited separately, giving rise to new combination of traits in the offspring after sexual reproduction. Sex determination in humans depends on which type of paternal chromosome, X or Y, fuses with the X chromosome of the egg. Evolutionary relationships are traced in the classification of organisms. Evolution can be traced out by the study of fossils also. Children, now I will ask you some questions and hope you will be able to answer them all correctly. So my first question is, how is the sex of a newborn child in humans determined? In humans, the nucleus of the cell has 23 pairs of chromosomes. Out of these, 2 chromosomes are sex chromosomes. These are X and X in females and X and Y in males. Children, an unfertilized egg always has a X chromosome in it. Now, if a X chromosome of the sperm fuses with X chromosome of the egg, a girl child will be produced. But children, if Y chromosome of the sperm fuses with the X chromosome of the egg, a boy child will be developed. My next question: what are homologous organs? And the correct answer is the organs which have the same basic structure but perform different functions are called homologous organs. Children, the next question for you is: what are acquired and inherited traits? And the correct answer is the acquired traits are the ones that a person develops during his or her lifetime. These are not passed from one generation to another. Inherited traits are present in a person since the time of his or her birth and are passed from one generation to another. Next question: what are fossils? The preserved traces of an organism are called fossils. And my last question, children: how can we find out that how old a fossil is? There are 2 methods by which we can find out how old a fossil is. In the first method, if we dig the earth and locate some fossils, the fossils which are closer to the surface of the earth are more recent ones from those we find in deeper layers. And the second method is by fossil dating. With this, we come to the end of our today's lesson, that was Heredity and Evolution. Hope children, you all enjoyed learning the lesson with me. We shall meet again with the next lesson. Till then, stay safe, stay healthy. Thank you.